about the types of centrifugation. That means types of centrifugal technique. We have already saw the types of centrifugation based on motors. Now we are going to see about the type of centrifugation based on the centrifugal techniques. So based on the centrifugal techniques means it is going to based on the sample separation. How does the sample and what can be the nature of sample to be separated? Based on this principle, the centrifugal techniques can be divided as preparative centrifugation and analytical centrifugation. In preparative centrifugation, where there is going to be a qualitative separation of the sample, where in the analytical both qualitative and quantitative can be possible. In preparative centrifugation, where there is not can be a more accurate type of centrifugation, but in the case of analytical technique it is going to be quantitative, so even a small changes in the sample or biomolecules can also be separated off. That means even a small molecular weight changes can also be accounted and can be undergo centrifugation, and also there is a separation of the sample even though there is a small molecular weight changes to be found. So based on the qualitative changes, a preparative centrifugation itself can be further classified as or further has a types of differential centrifugation and density gradient centrifugation. These two types of centrifugations are going to be based on the gradient to be used in the centrifugation under the sample. For the sample, there will be a different types of gradients can be used in the case of density gradient centrifugation. Based on the gradients and density of a sample, the separation can be found, where in the case of differential centrifugation, there will be only one gradient to be found. Due to the presence of only one gradient, the centrifugation can be taken place based on the RPM, where there is a different RPM can be used in the case of differential centrifugation, but in the case of density gradient centrifugation there is single RPM can be followed from the first to last of the centrifugation, but there will be a different gradient can be used so that different types of samples can be separated off in different gradients.